coming to the Carey Institute seminar series. For those of you who don't know me, I'm Jaslyn Hall. I'm a postdoc here at Carey, and I'd just like to thank you all for coming and to thank our virtual audience also for tuning in online. And I wanted to say that we will be saving questions to the end. For those of you who are in the virtual audience, if you're coming from far away or if it's your first time, feel free to add all of that information into the chat. And if you have a question for our seminar speaker, please put that into the Q&A so that we can review those at the end. And yeah, so it is my absolute pleasure to host and to introduce Dr. Sian Kou-Giesbrecht for this seminar specifically. Sian is an ecology or ecosystem scientist and modeler. She has a background in mathematics and environmental science for her undergraduate degree, so she's no stranger to the quantitative portions of ecology. And she also got her PhD from Columbia University. She was actually in the same PhD cohort as me, so we've known each other for about 7 years now. Sian studies the nitrogen cycle, and what I've always really admired about Sian's research is that she scales it really beautifully from looking at empirical plot and experimental data all the way up to global climate models, as you'll see very soon. So with that, I will let Sian take it away.
Thank you. Is this working? Yes. Okay. Thanks so much, Jazz. And thanks so much for inviting me here to present my research. It's really exciting to be here. And today I'm going to be talking to you about fueling the terrestrial carbon sink, nutrient cycling, and projecting climate change. So to start off with the really big picture, which I'm sorry if this is repetitive for some of you, but basically every year human activities emit 10.9 petograms of carbon per year into the atmosphere, primarily through burning fossil fuels. And about half of this stays in the atmosphere, and this causes the greenhouse effect. But about a third is sequestered by the terrestrial biosphere, in plant and in soil biomass, and this is what we call the terrestrial carbon sink. So the terrestrial carbon sink has a really huge impact on the amount of carbon dioxide that stays in the atmosphere and causes the greenhouse effect. But the terrestrial carbon sink needs nutrients such as nitrogen and phosphorus to fuel plant growth, because these nutrients form the building blocks of organisms alongside carbon. And nutrient limitation could be particularly important under elevated carbon dioxide concentrations, which stimulate plant growth and which correspondingly stimulate nutrient demand. So a really major challenge to our understanding of the terrestrial carbon sink is understanding the extent to which nutrients are going to limit the terrestrial carbon sink under future global change. So essentially, will the terrestrial carbon sink keep sequestering about a third of anthropogenic carbon dioxide emissions, or is this fraction going to increase or decrease given nutrient limitation? And so this is the question that I'm going to talk about today. So terrestrial biosphere models, or TBMs, are our key tool for simulating the terrestrial carbon sink. And what they do is they take our fundamental understanding of terrestrial ecosystems and they scale it to model terrestrial carbon sequestration at the global scale. So terrestrial biosphere models are the terrestrial components of Earth system models. So they interact with the oceanic and sometimes the cryospheric and the atmospheric components of Earth system models to model the global Earth system. And the Intergovernmental Panel on Climate Change uses an ensemble of different Earth system models. These are developed by different groups around the world, and they use these to project climate change. And an example of this would be the Canadian Earth System Model, which is the model that I work on at the Canadian Centre of Climate Modeling and Analysis. But nutrient cycling in these terrestrial biosphere models and in these Earth system models is still in its infancy. And this is despite this very empirically established importance of nutrient limitation of plant growth. So what this figure shows is the black line is the change in the terrestrial carbon sink over the past century and over the next century as simulated by an ensemble of the IPCC terrestrial biosphere models. But if we constrain this with empirical nutrient supply and stoichiometry estimates of nitrogen or of nitrogen and phosphorus, these are the red and blue lines, you can see that the terrestrial carbon sink at the end of the century, which we would normally expect to increase because of carbon dioxide fertilization of plant growth, is significantly decreased. And so this really kind of underlines the pivotal importance of nutrient cycling in terrestrial biosphere models. But these are just empirical constraints. These aren't actually based on an explicit representation of nutrient cycling in Earth system models. And that's because only about half of Earth system models have a representation of nitrogen cycling, and only one has a representation of phosphorus cycling. So incorporating and improving the representation of nitrogen cycling and phosphorus cycling in terrestrial biosphere models and in Earth system models is really important for addressing or for projecting the future terrestrial carbon sink under global change. So what I want to talk to you about today is just really how important it is for us to be able to kind of couple our empirical understanding of terrestrial ecosystems and these models in order to improve terrestrial biosphere models and to improve Earth system models and their projections of the terrestrial carbon sink and ultimately climate change. So before I kind of dive into terrestrial biosphere models and Earth system models, I want to first talk about some work that I've done on improving our fundamental understanding of nutrient cycling and nutrient losses in terrestrial ecosystems, and then move on to these big picture models. So a lot of my work has focused on biological nitrogen fixation. This is the dominant natural nitrogen supply to terrestrial ecosystems. So it's therefore extremely important for nitrogen cycling in terrestrial ecosystems. And biological nitrogen fixation is the conversion of nitrogen gas in the atmosphere into a plant available form of nitrogen by nitrogen fixing bacteria. And these nitrogen fixing bacteria can form a symbiotic relationship with plants in which they provide the plants with nitrogen in exchange for carbon, but they can also be free living nitrogen fixing bacteria such as soil microbes or lichens or some bryophytes. And biological nitrogen fixation is particularly important because it enables plants to overcome nitrogen limitations. So plants will upregulate biological nitrogen fixation in nitrogen limited conditions, and they will downregulate biological nitrogen fixation in nitrogen rich conditions. And that's why BNF is so important to understanding the extent that nutrients and nitrogen will limit the terrestrial carbon sink under global change. And I've been involved in a large-scale campaign of experimental studies, including one that I'm going to talk about later that was conducted very close to here in Black Rock Forest that essentially showed this downregulation or upregulation of biological nitrogen fixation under nitrogen-limited conditions. So in unfertilized treatments, nitrogen-fixing trees upregulated biological nitrogen fixation, whereas in higher nitrogen fertilization treatments, nitrogen-fixing trees downregulated biological nitrogen fixation. And this held across different symbiotic types as well as across latitudes. And also, why specifically do we care about nitrogen-fixing trees? It's because trees play a really key role, or nitrogen-fixing trees play a really key role in nitrogen supply to forests, which are really important for the terrestrial carbon sink, given the high carbon content of wood. So given that nitrogen-fixing trees can upregulate biological nitrogen fixation in these nitrogen-limited conditions, they stimulate carbon dioxide sequestration in plant and soil biomass by relieving nitrogen limitation of plant growth. Nitrogen fixing trees have also been suggested to enrich soil nitrogen through the turnover of their very nitrogen-rich tissues. And this has been suggested to relieve nitrogen limitation of neighboring trees by increasing soil nitrogen. And this would further stimulate carbon dioxide sequestration in plant and soil biomass. But some studies have also found that nitrogen fixing trees stimulate nitrous oxide emissions from soils. And this is again because they enrich soil nitrogen through that turnover of their nitrogen-rich litter. And this increases soil nitrogen, which essentially feeds the soil microbial processes that emit nitrous oxide. And nitrous oxide is a really potent greenhouse gas. It has a global warming potential that is 265 times that of carbon dioxide. So nitrogen fixing trees could be both stimulating nitrous oxide emissions from soils as well as carbon dioxide sequestration in plant and soil biomass. And this combination of these two effects, carbon dioxide sequestration on the one hand and nitrous oxide emissions on the other, this has been studied quite a bit for nitrogen fertilization in agriculture, but it hasn't really been studied in the context of biological nitrogen fixation. So to kind of get at how different nitrogen fixing trees can both stimulate carbon dioxide sequestration and nitrous oxide emissions from soils, first, we conducted a meta-analysis to examine the extent to which nitrogen fixing trees drive soil nitrous oxide emissions, because this was previously known from a handful of scattered studies. And what we found is that nitrogen fixing trees more than double soil nitrous oxide emissions relative to non-fixing trees. So they increase nitrous oxide emissions by 230% relative to non-fixing trees. And this held in both forests and grassland ecosystems, and it held in both managed forests as well as natural forests. So after establishing this widespread importance of how nitrogen fixing trees drive soil nitrous oxide emissions, we conducted an experimental study to look at the underlying mechanisms of how nitrogen fixing trees can drive both carbon dioxide sequestration and soil nitrous oxide emissions. And this field experiment was located in Black Rock Forest here in New York, and it consisted of these pairs of a nitrogen fixing tree as well as a non-fixing tree that were fertilized over 5 years in a control treatment and in a nitrogen fertilization treatment. And the nitrogen fixing tree was black locust or Robinia pseudoacacia. And what we did is we measured both carbon dioxide sequestration as well as nitrous oxide emissions that were driven by the nitrogen fixing tree as well as by the non-fixing tree. And this was a really big undertaking that would have been completely impossible without the help of two undergraduate students, Elsie and Gabriella. And what we found is first that nitrogen fixing trees promote carbon dioxide sequestration, which is shown here as relative biomass carbon growth rate in the unfertilized treatment, but not in the fertilized treatment. So in this figure, Robinia, or the nitrogen fixing tree, is shown in orange, and the non-fixing tree is shown in blue, and the unfertilized treatment is on the left, and the fertilized treatment is on the right. And the reason behind this is that nitrogen fixation, or the capacity of nitrogen fixing trees to fix nitrogen, allowed them to overcome nitrogen limitation in the unfertilized treatment, whereas the fertilized treatment wasn't nitrogen limited, so they exhibited similar biomass carbon growth rates. And second, what we found is that nitrogen fixing trees promote nitrous oxide emissions in both treatments. And this was because the nitrogen fixing trees did enrich soil nitrogen. So this figure shows nitrous oxide emissions under both the nitrogen fixing trees and the non-fixing trees, as well as inorganic soil nitrogen under both the nitrogen fixing trees and the non-fixing trees. And the nitrogen fixing trees were actually able to enrich the soil nitrogen through two different mechanisms. So the first is that nitrogen fixation allow the nitrogen fixing trees to maintain these really nitrogen-rich tissues, which is shown here as leaf nitrogen content. And these turn over and they decompose, and this enriches soil nitrogen. And the second is also that nitrogen fixing trees are able to satisfy most of their nitrogen demand through nitrogen fixation. That's shown here as the percent of leaf nitrogen that is derived from nitrogen fixation. And you can see that even in the fertilized treatment, this is still upwards of 60%. So essentially what this does is that it reduces soil nitrogen uptake, which leaves more nitrogen in the soil and effectively enriches soil nitrogen relative to the non-fixing tree. So overall, what this shows is that in the unfertilized treatment, nitrogen fixing trees promoted both carbon dioxide sequestration as well as soil nitrous oxide emissions relative to the non-fixing tree, whereas in the fertilized treatment, nitrogen fixing trees only promote nitrous oxide emissions relative to the non-fixing tree. So then after kind of getting at these underlying mechanisms of how nitrogen fixing trees drive both carbon dioxide sequestration and soil nitrous oxide emissions, we represented them in this exploratory analytical model to try and understand how these effects could change with global change. So the exploratory analytical model represented the pools and the fluxes of carbon and nitrogen in an ecosystem. And specifically, it represents carbon dioxide sequestration and nitrous oxide emissions of ecosystems with both nitrogen fixing trees and non-fixing trees. And what we were interested in exploring was intensifying nitrogen deposition. So nitrogen deposition is atmospheric input of nitrogen to soils. This can either be through precipitation or through aerosols. And importantly, nitrogen deposition has been increasing over the historical period. And this is primarily due to intensive agriculture, as well as fossil fuel burning, which emit nitrogen-based gases into the atmosphere. These circulate around and then get deposited on natural soils. So what we did is we took this simple exploratory analytical model and we simulated intensifying nitrogen deposition. And what we found is that under elevated nitrogen deposition, nitrogen fixing trees promote nitrous oxide emissions, but not carbon dioxide sequestration relative to non-fixing trees. And this is similar to the results of the field experiment. So this figure here shows how nitrogen fixing trees either promote carbon dioxide sequestration, which is in blue, or nitrous oxide emissions, which are in red, under low nitrogen deposition rates, as well as under high future projected nitrogen deposition rates. And you can see that under low or current nitrogen deposition rates, nitrogen fixing trees promote carbon dioxide sequestration relative to non-fixing trees and slightly promote nitrous oxide emissions, whereas under high nitrogen deposition rate, these trees aren't nitrogen limited, so they are solely promoting nitrous oxide emissions relative to non-fixing trees. So what this suggests is that nitrogen fixing trees could be really important for accounting for soil nitrous oxide emissions in natural ecosystems, especially in areas that are receiving high nitrogen deposition rates. And nitrogen fixing trees are particularly important because they are often promoted as being planted during reforestation. This is because of two different reasons. So first, they grow really rapidly on nitrogen poor soil, and also because they have this proposed potential to relieve nitrogen limitation of neighboring trees. So as an example of this, Ingas have been promoted is a good tree for reforestation projects in the Amazon. But essentially what our work has shown is through multiple lines of evidence that nitrogen-fixing trees could also be driving soil nitrous oxide emissions. This could potentially somewhat offset carbon dioxide sequestration from reforestation, and really needs more research into its accounting. Also, this proposed potential that nitrogen-fixing trees could relieve nitrogen limitation of neighboring trees might not actually hold as well. So we conducted an analysis of the US FIA Forest Inventory and Analysis Database across the coterminous United States. And what this showed is that actually nitrogen-fixing trees have no net facilitation effect on neighbor growth rate. They had a mildly negative effect on neighbor growth rate. However, they did have a positive effect on neighbor recruitment rate, and they had a negative effect on neighbor survival rate. So nitrogen-fixing trees could be promoting forest turnover. But overall, this is kind of just another caveat in this potential for nitrogen-fixing trees during planting nitrogen-fixing trees during reforestation projects. So now I want to move on to talking about modeling and terrestrial biosphere modeling and Earth system modeling. And so I'm currently working on how we represent nitrogen cycling and nitrogen fixing trees and biological nitrogen fixation in general in the Canadian land surface scheme, including biogeochemical cycles, or CLASSIC, which is the terrestrial biosphere model component of the Canadian Earth system model, which contributes to the IPCC assessment reports. So biological nitrogen fixation has been identified as this really central challenge to representing nutrient cycling in terrestrial biosphere models. First, because it is the dominant natural nitrogen supply to terrestrial ecosystems, so is therefore extremely important, but also because this regulation of symbiotic biological nitrogen fixation by plant nitrogen limitation is a very complex feedback that can be challenging to represent. However, it is really important for understanding this question, to what extent will nutrients limit the terrestrial carbon sink under global change, because it could potentially give plants the capacity to respond to elevated nitrogen limitation under carbon dioxide fertilization. So normally in terrestrial biosphere models and Earth system models, we represent nitrogen fixation phenomenologically as a function of plant biomass. So either as a function of net primary productivity or as a function of evapotranspiration, which are both empirically derived. And the logic behind this is that if you increase plant biomass, you will inevitably increase nitrogen fixing plant biomass, which means that you should have more biological nitrogen fixation. But the problem with this is that it doesn't really capture this regulation of biological nitrogen fixation by nitrogen limitation, because if you decrease nitrogen limitation, you're going to increase plant biomass. So if you calculate biological nitrogen fixation as a function of plant biomass, you'll get increasing biological nitrogen fixation with decreasing nitrogen limitation, rather than decreasing biological nitrogen fixation with decreasing nitrogen limitation, which we have empirically established. So this really illustrates kind of a large problem with terrestrial biosphere models, which is that we tend to represent a lot of these functions phenomenologically rather than looking at their controls and understanding their mechanistic drivers. So to address this, we developed a new representation of biological nitrogen fixation in CLASSIC, which I won't really get into the weeds of, but essentially we have biological nitrogen fixation represented as a function of nitrogen limitation of plant growth. And we also developed a separate function, free-living biological nitrogen fixation representation that is independent from nitrogen limitation of plant growth. So before I go into those results, I want to briefly explain how a terrestrial biosphere model works. Essentially, a terrestrial biosphere model is fed a set of inputs. These include carbon dioxide, temperature, precipitation, other meteorological forcings, nitrogen deposition, land cover, so both natural vegetation area and crop area, as well as nitrogen fertilizer application. And a terrestrial biosphere model is given this set of inputs, and it simulates various aspects of the Earth system. And I've highlighted these specific inputs because these are global change drivers that have changed over the historical period and that are also going to change in the future. So you can see carbon dioxide has increased, temperature has increased, precipitation has become a lot more variable, nitrogen deposition has really increased, natural vegetation area has decreased while crop area has increased, and nitrogen fertilizer application has increased over the historical period. So Classic is given this set of inputs, and it simulates energy, water, carbon, and nitrogen fluxes and pools. So this is a simplified representation of the way that Classic represents all of these different fluxes and pools. And this really kind of gets at the complexity that underlies a terrestrial biosphere model. So with our new mechanistic representation of biological nitrogen fixation, Classic is able to represent the current global total of biological nitrogen fixation, where Classic is indicated here in green and observations are indicated here in gray. The two colors represent symbiotic biological nitrogen fixation as well as free-living biological nitrogen fixation. And you can see that Classic is really similar to the 5 other IPCC terrestrial biosphere models that represent biological nitrogen fixation. The problem here is that in order to really evaluate the representation of a new process that's incorporated into a terrestrial biosphere model, it's not really good enough to look at just the global total estimate. It's really important to look at, well, specifically for biological nitrogen fixation, the response to varying nitrogen limitation, but also the response of these new processes to global change, especially within the context of empirically established information. So to do this, we simulated 2 different fertilization experiments, or 2 different global change experiments. And the first was a carbon dioxide fertilization experiment. So essentially what we did is we just increased carbon dioxide concentrations very suddenly across the globe. And the first thing that we expect is for plant carbon sequestration to increase, because carbon dioxide fertilization should stimulate photosynthesis, which should increase plant growth. And we also expect biological nitrogen fixation to increase. And this is because that stimulated photosynthesis should increase nitrogen limitation, which should increase biological nitrogen fixation. And here, these gray lines are results from meta-analyses of experimental studies, primarily FACE experiments. And so what we find is that Classic is able to reproduce both of these responses. So Classic is indicated here in green, and the other IPCC terrestrial biosphere models are indicated here in orange. The second global change experiment that we simulated was a nitrogen fertilization experiment. And this could be representative of intensifying nitrogen deposition. So essentially, we just took nitrogen deposition rates and increased them across the globe. And what we expect again is that plant carbon sequestration should increase, because increasing nitrogen fertilization should relieve nitrogen limitation of plant growth. And we also expect biological nitrogen fixation to decrease. And this is because with increasing nitrogen fertilization, this should relieve nitrogen limitation, and this should decrease biological nitrogen fixation. And again, the gray lines are meta-analyses of experimental studies. And so what we find is that classic was able to reproduce both of these responses. And what's really interesting here is that because most of these other IPCC terrestrial biosphere models use a phenomenological representation of biological nitrogen fixation, they predict increasing biological nitrogen fixation with nitrogen fertilization, because again, this decreases nitrogen limitation, which should increase biological nitrogen fixation, rather than the empirically established decrease in biological nitrogen fixation with increasing nitrogen fertilization. So after developing a representation of biological nitrogen fixation as the dominant natural nitrogen supply, we can really look at how a representation of nitrogen cycling influences projections using terrestrial biosphere models of the terrestrial carbon sink. So to look explicitly at the role of nitrogen cycling, we compared two different versions of classic. So classic CN, which has this representation of nitrogen fixation and of nitrogen cycling, and classic C, which does not have a representation of nitrogen cycling. And again, just as a reminder, only about half of the IPCC terrestrial biosphere models do have a representation of nitrogen cycling. So this figure here shows net terrestrial carbon sequestration of the entire terrestrial biosphere over the historical period. And classic CN is indicated by the dotted lines, and classic C is indicated by the salt lines. These yellow bars are from atmospheric inversions of carbon dioxide. So you can see that both classic C and classic CN are able to reproduce the historical terrestrial carbon sink, which is currently taking in about a third of anthropogenic carbon dioxide emissions. And this is true for pretty much all terrestrial biosphere models. So this is a figure from the most recent global carbon budget. Each of these green lines is a different IPCC terrestrial biosphere model. So they're actually all pretty similar, and they're all simulating that the terrestrial biosphere is taking in about a third of anthropogenic carbon dioxide emissions. But what's kind of weird is that only half of those terrestrial biosphere models have a nitrogen cycle and half of them don't. So to kind of figure out what is going on, we looked at each of the global change drivers that are underlying this terrestrial carbon sink independently. So the terrestrial carbon sink is a result of carbon dioxide, climate, nitrogen deposition, and land use. So we looked at each of these individually. So this figure shows how each of those different global change drivers contributed to the terrestrial carbon sink over the historical period. And when you compare classic CN, which is indicated by the white bar, and classic C, which is indicated by the gray bar, when you compare them with all global change drivers acting concurrently, it's a pretty similar result. And that's why they're both able to reproduce the historical terrestrial carbon sink. But if you look at each of the global change drivers individually, the differences between classic C and classic CN become quite apparent. And I'm just going to look at carbon dioxide and nitrogen deposition here. So over the historical period, carbon dioxide has increased, and this has driven increasing terrestrial carbon sequestration. But you can see that the amount that this has stimulated carbon dioxide sequestration is pretty different between classic C, which is gray here, and classic CN, which is white. And I'm going to come back to that in a second. But the second big thing here is that nitrogen deposition has also increased over the historical period. This has stimulated the terrestrial carbon sink, but this isn't represented in classic C. This is only represented in classic CN, so that's why there isn't a gray bar over there. So overall, what this is showing is that in order for classic C to reproduce the historical terrestrial carbon sink, the contribution of carbon dioxide to the historical terrestrial carbon sink is pretty much artificially increased in order to compensate for the contribution of nitrogen deposition, which is not represented. And you can't really see that when you look at all of the global change drivers acting at the same time, but you can if you separate each of them out. And specifically what was happening here is the sensitivity of photosynthesis to carbon dioxide was increased in classic C in order to increase the net terrestrial carbon sequestration due to carbon dioxide. And this isn't unique to classic. So essentially all terrestrial biosphere models are tuned in order to reproduce the historical terrestrial carbon sink. You can't contribute to the global carbon budget if you aren't able to reproduce this historical terrestrial carbon sink. And most of them are tuned through that parameter, which dictates the sensitivity of photosynthesis to carbon dioxide. But the problem with this is that it introduces these compensatory effects between carbon dioxide on the one hand and nitrogen deposition on the other, because the terrestrial carbon sequestration that is due to carbon dioxide has to be artificially increased to compensate for the fact that the terrestrial carbon sequestration due to nitrogen deposition is not simulated. So this kind of begs the question of what is going to happen in the future, and how is this going to influence future projections of the terrestrial carbon sink? So the shared socioeconomic pathways are a set of alternative future scenarios. These depend on our socioeconomic development over the next century, and they range from this really wonderful sustainability scenario where we figure everything out to a fossil fuel development scenario where we don't figure anything out. And these are the framework that is used by the IPCC. And each of these different scenarios outlines different trajectories for all of the global change drivers that I just described. So carbon dioxide, meteorological forcings, nitrogen deposition, land cover, and nitrogen fertilizer application. But what's really important here is that these trajectories in the future diverge from their historical trajectories. So over the historical period, carbon dioxide increased and nitrogen deposition increased. In the future, carbon dioxide tends to increase in many of these scenarios. So SSP5 is indicated in red and SSP3 is indicated in gold. But nitrogen deposition actually plateaus or decreases in all of these future scenarios. So what this figure shows is net terrestrial carbon sequestration as predicted by classic Sian and by classic C. And again, both of them are able to reproduce the historical terrestrial carbon sink, but in the future, their projections really diverge. So in this worst case future scenario, SSP5, the projections by classic CN are 64% lower than the projections by classic C. So basically, a terrestrial biosphere model that doesn't represent nitrogen cycling, that is able to reproduce the historical terrestrial carbon sink, is going to overestimate the future terrestrial carbon sink. And it's easy to say that this is just because, oh, well, classic CN has nitrogen limitation represented, so obviously it's just going to be lower. But it's actually not really that simple, because both of them are able to reproduce the historical terrestrial carbon sink. Nitrogen limitation affects the historical terrestrial carbon sink. It should be implicitly baked in there. But what's really underlying this is these compensatory effects between carbon dioxide and nitrogen deposition. So the terrestrial biosphere models have all been tuned to the historical trajectories of both carbon dioxide and nitrogen deposition, which have both increased simultaneously over the historical period, but then these diverge in the future. And that's essentially what leads to this huge discrepancy between models with nitrogen cycling and models without nitrogen cycling. And this clearly will have really big implications, especially when these are put into a coupled air system model for projecting atmospheric carbon dioxide concentrations and climate change. Nitrogen cycling is also really important because enriched nitrogen causes all these really important nitrogen losses from terrestrial ecosystems. So for example, I talked about nitrous oxide emissions, nitrous oxide being a really important greenhouse gas, but also NOx emissions from soils can cause smog and air pollution, and nitrogen leaching into water causes eutrophication. And these are primarily driven by nitrogen deposition and nitrogen fertilization, which is another aspect of the Earth system that we can simulate with an explicit representation of nitrogen cycling. And these simulations of the future scenarios show that all of these are going to increase in the future, primarily driven by increasing nitrogen fertilization, with really important socioeconomic consequences. So the final thing that I want to talk about today is, aside from classic, how do terrestrial biosphere models and Earth system models in general represent nitrogen cycling? So in the IPCC reports, every result is from an ensemble of different terrestrial biosphere models and Earth system models. And for carbon cycling, the main group behind this is the Global Carbon Project. And they report an annual global carbon budget, and this ultimately informs the IPCC assessment reports. And this includes an estimate of the terrestrial carbon sink, which is shown here. This is from an ensemble of Earth system models and an ensemble of terrestrial biosphere models, and in combination with atmospheric carbon dioxide inversions is how we know that over the past 50 years or so, the terrestrial biosphere has been sequestering a third of anthropogenic carbon dioxide emissions. And these different terrestrial biosphere models are really rigorously evaluated with respect to carbon cycling. So we compare their model output to observation-based data sets of several different variables, which are shown here on the left. The models are shown here at the top, and their score in their ability to reproduce those observation-based data set is shown here. So an example of this would be vegetation biomass. We have an observation or a product that is from remote sensing and from these in situ plots, and you can compare that to simulated model output to derive a score for how well the model was able to reproduce it. But the problem here is that none of these terrestrial biosphere models are evaluated with respect to nitrogen cycling, even though about half of them now have a representation of nitrogen cycling. And this is in part because nitrogen cycling is relatively new in these terrestrial biosphere models, but is also because we have a general kind of lack of good global nitrogen cycling products. So what we sought out to do was to evaluate nitrogen cycling in the terrestrial biosphere models that contribute to the IPCC that actually have a representation of nitrogen cycling. But to do this, first we had to kind of generate these new global nitrogen cycling data products. So the first one that we looked at was biological nitrogen fixation. This product was derived from a natural nitrogen fixation meta-analysis in combination with agricultural nitrogen fixation rates and nitrogen fixing crop coverage. The second was vegetation carbon to nitrogen ratio, which we derived from the tri-plant trait database using measurements of tissue carbon, tissue nitrogen, and different tissue allocations in combination with the International Geosphere Biosphere Program BioMap. And the final data product was a soil carbon to nitrogen ratio product, which we derived using soil carbon, nitrogen, and bulk density data from the World Soil Information Service. So this figure shows the latitudinal distribution of the IPCC terrestrial biosphere models in the blue-green colors in comparison to our new observation nitrogen product, which is indicated by the pink colors. And so what you can see is that the IPCC terrestrial biosphere models are really overestimating tropical biological nitrogen fixation, and they're underestimating temperate biological nitrogen fixation, assuming the validity of that observation-based product. This figure shows vegetation carbon to nitrogen ratios. Again, the models are bluey green and the data product is pink. And you can see that all of the models are overestimating vegetation carbon to nitrogen ratio, but they are able to capture the latitudinal pattern. And this final figure shows soil carbon to nitrogen ratio. And you can see that the models are overestimating soil carbon to nitrogen ratios across the latitudes, and they're not able to capture the latitudinal pattern whatsoever. They're simulating a pretty constant carbon to nitrogen ratio across latitudes. And in addition to that, the different terrestrial biosphere models are also exhibiting really huge variability across their simulations of important nitrogen cycling pools and fluxes. So here, each of these different lines is a different model. And this is showing vegetation nitrogen, litter nitrogen, soil nitrogen, and biological nitrogen fixation. And so, and this shows change since the historical period. And you can see that there are really different magnitudes and trends going on. But despite this huge variation in their simulations of nitrogen cycling, again, all of these models are able to simulate the terrestrial carbon sink. So each of these lines is one of those different terrestrial biosphere models. The boxes are, again, the atmospheric inversions of carbon dioxide. And so you can really see that there's a pretty big disconnect between the nitrogen cycle simulations and the carbon cycle simulations. So these underlying nitrogen processes that determine nitrogen limitation of plant growth and terrestrial carbon sequestration operate really differently across the terrestrial biosphere models that represent nitrogen cycling. So overall, what this shows is that the half of the IPCC TBMs that represent nitrogen cycling overestimated tropical biological nitrogen fixation, overestimated vegetation carbon to nitrogen ratios, and overestimated soil carbon to nitrogen ratios. So they're overestimating both nitrogen supply to the terrestrial biosphere, and they're also overestimating the amount of carbon that can be sequestered per unit nitrogen, which could be pretty important for projections of the future terrestrial carbon sink, especially under elevated carbon dioxide concentrations and global change. And that's to say nothing of the other half of the IPCC terrestrial biosphere models that don't represent nitrogen cycling whatsoever. So with that, I kind of just wanted to emphasize that we often have this like big disconnect between empiricists over here and the modelers over here. So it's really important to, really represent well these empirically established underlying mechanisms for the terrestrial ecosystem processes that we are trying to capture in these models to then ultimately make these projections of the terrestrial biosphere, the terrestrial carbon sink, and climate change. So that I would like to thank various people and funding sources and take any questions.
Thank you very much, Sian. That was very interesting. So yeah, we will take questions, I think, from the audience, a couple. And then for the virtual audience, if you have questions, please do add them into the Q&A and we will read them aloud.
Sarah?
Like a lot of, so you were, the empirical work that you did at Blackhawk, you found that the fixers continue to fix the majority of the nitrogen, or get the majority of the nitrogen from the station to the money or fertilized, and that then it has the high emissions. Getting done, and that makes sense, although I'm curious, because that's, well, so there's the whole idea of some nitrogen fixings doing or treating doing down in vegetation as like the theory of development exponentially, where if there's a lot of nitrogen available, then it'll down negative fixation. And you found some evidence of that for the
Mhm.
when you added nitrogen, but still like they were 50 out, so it wasn't like complete down-regulation. In fact, it was like only a little bit of down-regulation. And I'm curious if you thought, like, if why was it that they didn't down-regulate the whole thing? And especially in the context of silver finding and Kou-Giesbrecht, this found that rubidium does down-regulate when they don't need it. And so that seems to be different between, and her work is like immature trees. So I'm wondering if there's something about these like 5-year-old trees that they're not actually fully down-regulating. And then the other part is then like scaling from like that finding to, you know, other terrestrial ecosystems and projecting nitrous oxide emissions, and especially for talking about where we know that the majority of trees actually do fully down-regulate or almost completely down-regulate vegetation. If there would be so much nitrous oxide emissions in other regions of the tropics as what we have projected.
Yeah, that's a really great question. So, oh yeah, okay. Uh-oh. Oh no. So the question was, I guess part of it was like, do nitrogen fixing plants, like we observed that they partially downregulated in our experiment in Black Rock, and there were also some accompanying experiments there. There was one in Hawaii and one in Oregon, but why didn't they completely downregulate? Whereas there have been observations that they would completely downregulate. And that's a really great question. And that was a surprising result. And I think that like you raised a really good point that it probably is just that they're young. So like, whereas in a mature forest, if they're not nitrogen limited, they might completely downregulate the fact that they're pretty young, 5 years old, although we were fertilizing them with so much nitrogen, 100 kilograms of nitrogen per hectare per year, they didn't completely downregulate. And then I guess how this would influence nitrous oxide emissions, that's another really great point, that the nitrous oxide emissions aren't so much an effect of the nitrogen fixation, they're effect of the nitrogen status of the system. So if plants are super nitrogen limited, they'll take up a ton of nitrogen, so there won't be a lot of nitrogen left in the soil to be lost through either nitrous oxide emissions or NOx emissions or ammonia emissions or nitrogen leaching. So I think a challenge with this is that because it was experimental and rather than looking at like a non-nitrogen limited forest versus a non-nitrogen limited, not nitrogen fertilized forest, we were looking at a fertilized forest. So the overwhelming driver of nitrous oxide emissions was the fact that we were fertilizing them with 100 kilograms of nitrogen per hectare per year. And because nitrous oxide emissions are so temporally and spatially variable, it's like a little bit hard to like parse that out. But I think that's a really good point, that especially in a tropical forest, which is a lot less nitrogen limited than a temperate forest such as Black Rock, nitrous oxide emissions could be much more relevant. I don't think there'd be like a clear direct effect of nitrogen fixation driving nitrous oxide emissions. It's more so like the ultimate ecosystem effects that they have on soil nitrogen. But that definitely could be much more important in a tropical forest. And that's especially true if you look at just like nitrous oxide estimates from temperate forests. They're really, really low in comparison to tropical forests. Yeah. Yes. Oh, sorry.
Thanks for a really great talk. That was fun.
Thanks.
And I was really interested. I really enjoyed the section where you talked about comparing models that included a nitrogen cycle versus not. And it was quite striking to see what an effect that had on outcomes. But there are also a whole litany of other processes that TBMs don't include. So forest demographics, for example, or disturbances in spatially explicit ways. And so in your mind, how do you compare the relative importance of investing in building out mechanistic nitrogen cycling versus all of those others that would also take a substantial amount of investment and effort in terms of like really trying to better constrain our projections in the future of carbon taking?
Yeah, that's a really great question. That like with all modeling, you kind of run into this like, is it worth the complexity? Because every time you add in a new process, you add in parameters that are unconstrained and everything gets a little bit funkier. So trying to like kind of justify which processes really need to be included is kind of this unanswerable question. I think that forest demographics is a really important aspect to include. The problem with forest demographics is the computational costs, whereas with nitrogen cycling, it's a little bit easier to like include, whereas like if you start including stands of various ages within, I mean, we're simulating at like a 1 degree by 1 degree grid cell. So if you try and like divide that into stands of different ages, it's just going to be a mess eventually. Although there have been like developments in the ways that we are able to represent that, like with the Fates model or with perfect plasticity approximation. I think that, I mean, maybe I'm just biased, but I do think that like nitrogen limitation and phosphorus limitation are super important, especially in the context of carbon dioxide fertilization. But I do think that like fire and stand dynamics are going to be like the next big push, especially with like fire and the way that that structures forest ages. I feel like that's the next big push that's coming. Shall?
And end up position, and I don't know if that's right question, but if you break it down by grid cells, you kind of see that relationship like even stronger, like in some of the grid cells that have been historical low end up position, or I don't know, maybe there's other factors.
Yeah, that's a great question. And I'm trying to, so actually one of the interesting results that we found by looking at it by grid cell was the effect, oh, sorry. Oh, okay, sorry. So yeah, the question was, I guess, if you look at different grid cells, which I'm assuming, like if you think about a latitudinal gradient, let's say, are there differences in the carbon dioxide effect versus the nitrogen deposition effect? And I don't remember too well. So we do observe, like, and this is primarily from the global nitrogen fertilization experiment, that you do see that the higher latitudes are more nitrogen limited and the lower latitudes. A caveat to this is that there isn't phosphorus cycling. So phosphorus limitation isn't represented. What was really interesting was, and I didn't show this, was the effects of climate between the two of them. And this differed quite a bit versus in the high latitude forest or high latitude ecosystems versus the low latitude ecosystems, which is that nitrogen mineralization gets stimulated with increasing temperature at the high latitude ecosystems. So actually, like the one with the nitrogen cycle, classic Sian, has more terrestrial carbon sequestration than classic C at the high latitude ecosystems because nitrogen mineralization gets stimulated. I believe that the effects of carbon dioxide were stronger at the lower latitudes just because of a lack of nitrogen limitation, but also not having phosphorus limitation is a big caveat to that.
I have a question from our virtual audience. Peter Severance has asked, how do soil carbon and nitrogen losses to rivers via groundwater and surface flows play into these TBM models?
Yeah, that's a great question. So there is nitrogen leaching to water bodies, but it kind of just ends there, which is unfortunate. So you can, like, I think I showed this at one point, that you can measure, like you can project how much nitrogen is lost to water bodies, but its effect on like actually like the representation of ocean processes or freshwater process, there's like a disconnect there actually. So it doesn't actually feed into the rest of the other parts of the model quite yet. That's true for the model for classic. It might be a little bit more complex in other models.
Your black rock study, in your plot showing the fertilized and unfertilized responses, is it a significant difference between that raise in the N fixed trees without fertilizer compared to both your N fixers and, like, I guess in the fertilized plots you saw the N fixers and the non-fixers to be the same, but was the unfertilized plots, were the N fixers even higher growth than N fixers in the exerting addition plots, or is that?
They were not significantly higher, but they were like, they trended to be higher, but they were like pretty much the same. Yeah.
I was curious if they were why that was the case.
Yeah, they were pretty similar. And we explicitly chose the paired non-fixing tree river birch because they exhibit like similar growth rates to Robinia. So they were like a decent comparison to make.
We have time for maybe one more question.
I have, it's not directly related to nitrogen, but given your expertise with working with many different terrestrial biosphere models, I've done like informal comparisons to think about downscale and climate at regional scale. So like, what is the best set of TBMs for representing climate in Alaska, for example? Do you find that at a regional level, the countries do the best in their own country? Like, is CAN-ESM best at creating regional climate in Canada and Alaska versus other models, or is it totally just all?
That's a good question. So I guess in theory, like CAN-ESM as like a contributor to the IPCC, we're supposed to be a global models, but there's kind of been like a subset group that has broken off that have like decided to kind of exclusively focus on like Canada and to break it down into a 0.02 degree grid cell and to try and like really focus in on the boreal forests. But I would say that that's been like a recent push of the past 5 years. And generally they do try to focus on the global scale. I think that often the way that these Earth system modeling groups are structured is you have like a land group, you have an atmosphere group, you have a cryosphere group, and you have an ocean group. So often those like, I think the land has maybe, again, bias, but the land has like more obvious like distinctions between different biomes, whereas like the atmosphere and like the ocean groups, it's more like continuous across the entire globe. So I think generally these groups tend to be like structured at the global scale.
Well, thank you very much, Sian, for presenting your work, and thank you all for joining us. And yeah, we'll see you next week.
Okay.